So this video is an introduction to transport across the membrane. So the membrane, as we said, is very important in regulating the internal environment as compared to the external environment of the cell. And because of the composition of the membrane, that's pretty easy to do. Remember that these head groups, the phosphate head groups, are hydrophilic. The fatty acid tails are highly hydrophobic, and that means that not very many things can pass through easily. So the requirements in order to be able to pass through easily are that the molecule be nonpolar, that it be very small, and that it be uncharged. And the reality is that eliminates a huge number of biological molecules. In fact, the only important molecules that seem to be able to pass through easily through the membrane are oxygen and carbon dioxide. So that's nice, but that's hardly anything for the cell. The cell needs a lot more. So what about the rest of the stuff? So there are two sort of major types of transport. There's passive transport, which requires no energy, and active transport, which requires energy to move molecules across the membrane. Passive transport can be broken down into diffusion for things like oxygen and carbon dioxide that can move easily, or facilitated diffusion. In some cases, in fact in most cases, molecules need a pathway through the membrane. They can't just make their way in between those phospholipids. And there are a few different ways that this can happen. So we're going to look first at this process of passive transport and look at some examples. So one really important molecule that passively moves through the membrane is water. It used to be thought that water just moved easily between those phospholipids, but based on what we know about the chemistry, that doesn't really make sense. And not too long ago, there was a protein discovered that actually provides a way for water to move much more easily through the membrane, and that protein is called aquaporin. This is a great example of what's called a channel protein. A channel protein is an integral membrane protein, and what it does is simply provide a tube for the molecule of interest to move through the membrane. And these channel proteins are selective based on potentially the size of the molecule. In some cases, they're selective based on the charge or the shape, but aquaporin is quite picky for just allowing water to move through. And again, the molecule is moving without any energy input. So why would water move across the membrane? Well, molecules flow to where there are fewer of that molecule. And that difference in concentration is referred to as a concentration gradient. So for example, if we have a situation where we have some molecules of dye in water and some plain water, and a membrane in between them, what we call a selectively permeable membrane, meaning that some things can move through and some things can't. Those dye molecules will tend to spread out. If you remember from chemistry, this is the idea of increasing entropy, increasing randomness in the situation. Those dye molecules will spread out until the concentration is as equal as possible. And they've moved through the membrane in order to do that. So this occurs even when there are multiple molecules involved. We've got one solute on one side, a different solute on the other side, and each of these solutes will move to a point of equilibrium so that the concentration of each is balanced on each side. Now there are other factors that affect exactly how much of each solute moves to each side, but you get the basic idea. So what does this have to do with water? Well, there are lots of situations where cells have different concentrations of solutes than their surroundings. And water is going to move from one side of the cell membrane to the other. So let's look at how that works. So here we've got a situation where we have a semi-permeable membrane. And that semi-permeable membrane has a high concentration of sugar on one side and a lower concentration of sugar on the other. Well, the sugar molecules in this case are too large to move across the membrane themselves. So you would imagine that the sugar would just move from one side to the other to equalize these concentrations. But that's not possible in this case. But what we can consider is that if there's a high concentration of sugar in this volume, there's actually a lower concentration of water. There are fewer water molecules per volume on this side than there are on the other side. So the water molecules, which can move through this membrane, are going to move to equalize the concentration. They're going to move from where there's more water molecules to where there are fewer water molecules. That passive movement of water down its concentration gradient is called osmosis. So water will move from where the concentration of water is high down its concentration gradient so that there are more equal concentrations of water on each side. And what you notice is that the volume over here is actually going to increase. Now, at some point, the physical pressure of this amount of water pushing down is going to start to squeeze those water molecules back the other way. So as I mentioned, there are multiple factors that control exactly how much of a molecule moves from one side to the other, but you definitely will get water movement. And this is really important for cells. So cells might be in a situation where the concentration of solutes is greater on the outside of the cell than on the inside. Water is going to move out in that case, and that causes the cell to shrink up because it's losing water. That turns out to be very bad for plants. It's what causes plants to wilt because their cells shrink and the plant falls over. Solutions where you have more solute on the outside than on the inside of the cell are referred to as hypertonic solutions. Solutions that where the same concentration of solutes is on the inside of the cell as the outside are referred to as isotonic solutions. Cells do well in those situations. The movement of water is equal across the two sides of the membrane. Situations where the cell has more solutes than its surroundings, that solution, that surrounding solution, is referred to as hypotonic to the cell. That tends to cause the cell to swell because water is flowing in an effort to equalize the concentrations of water on both sides. And this can actually cause lysis, or essentially explosion of the cell in. So organisms have evolved mechanisms of regulating the flow of water from one side to the other. And this is a paramecium caudatum, and it has this structure called a contractile vacuole. So this organism lives in a hypotonic solution. So water is constantly flowing into the organism, and it has to get rid of that water somehow. It collects that excess water in this contractile vacuole and literally pumps it back out. Now, every organism has evolved its own mechanisms for surviving and regulating its water balance in its particular environment. So just as a reminder, we've been talking about channel proteins, proteins that essentially make a tube from one side of the membrane to the other so that a molecule can flow through. And we've talked about aquaporins, which are the channel protein that allow water to move through the membrane. No energy involved. Another way to allow facilitated diffusion is what's called a carrier protein. Carrier proteins aren't just an open tube, they have a sidedness to them, and they change shape to allow the molecule to move from one side to the other, but again, they require no energy. This is all passive transport. So what about active transport? Water's all good that it's moving down its concentration gradient, but if we need to move something against its concentration gradient, we're going to need to put in some energy. So how are we going to do that? So here's a classic example of an active transport system. This is called the sodium-potassium pump. And we're going to go through these steps in detail, but the basic idea is that this molecule, which is an integral membrane protein, again, is going to take 3 sodiums. It's going to push those sodiums against their concentration gradient towards the outside of the cell. And it's going to take 2 potassiums, push those potassiums against their concentration gradient towards the inside of the cell, and finish. And this is all going to require energy. So what are the steps in this process? Well, the first step is for this sodium-potassium pump to extract, essentially grab the sodiums, even though their concentration is low. This protein has a very high affinity attraction for the sodiums. It picks up 3 sodium molecules. Now that it has 3 sodium molecules, it needs to get some energy to move those sodium molecules against their concentration gradient. So that energy is going to come from ATP. So the ATP is going to be broken down into ADP and the phosphate. That phosphate is attached now to our sodium-potassium pump. Now, if you think back to what happens to enzymes when they have a phosphate group attached to them, it shouldn't surprise you that this molecule is going to change shape and become activated as a result of having this phosphate attached. So the shape change means that the sodium-potassium pump is now open to the outside, and those sodiums are going to be released because the affinity, the attraction of the sodium-potassium pump for the sodiums is much lower in this conformation. Now the sodium-potassium pump can grab onto the small amount of potassium that's in this extracellular matrix. The potassium binding causes the release of the phosphate, and if you remember, no phosphate means open to the inside. So that pump is now open to the inside, the affinity for the potassiums decreases, and those potassiums are released into the inside of the cell. Now the result of this is not just that we've increased the concentration of sodium outside and increased the concentration of potassium inside, but because these molecules are charged, we've also now got a lot more pluses on the outside than the inside because this was an uneven exchange. This creates what's called a membrane potential. It's literally an electrical voltage or force across the membrane. And this force is important for lots of processes in the cell. We'll talk about it especially in relation to signaling in neurons and in immune cells. And the sodium-potassium pump is referred to as an electrogenic pump for that reason, because it creates that electrical difference between the two sides of the membrane. Plants do this a little bit differently. Their main electrogenic pump is actually a proton pump. So instead of using sodium-potassium, they're using hydrogen ions. Hydrogen ions are pumped to the outside of the cell so that they're higher concentration outside, again using ATP. Because you have this imbalance in charges, that creates an electrical force. There's an attraction for positive charged molecules to move towards the inside of the cell. And you can use that force then to transport other things. So you can imagine as the proton moves down its concentration gradient and down the electrical gradient, the energy released in that process can be used to co-transport molecules that the cell wants, such as sucrose, which is an energy source. So this is a common process in plant cells. What if the cell needs to take in more than one molecule at a time? You can imagine just doing one at a time might be pretty slow. So the cell has a few different mechanisms for taking in larger volumes of stuff. The first of those mechanisms is called phagocytosis. This is a situation where the cell is taking in sort of chunks or particles of material. Not every cell can do this, but the basic idea is that the membrane sort of folds around the particle, and that particle ends up in a vacuole or a small membrane-bound compartment within the cell. Another mechanism, if you don't need to take in a huge particle, is what's called pinocytosis, where you're taking in many molecules at once. And basically, again, the membrane folds inwards, taking in lots of molecules. In many cases, that folding process is helped along by a coat protein on the inside of the membrane that helps the membrane make this shape. Because remember, our membrane is quite flexible. That's all well and good if you want to just take in random molecules that happen to be nearby. If you want to be picky about what you're taking in, the cell uses receptor-mediated endocytosis. So same basic idea, but in this case, the cell has receptors binding to molecules that the cell especially wants to pick out of the mixture in the area. So those ligands bind to the receptors are attached to the membrane. When the membrane pinches off, you've got a high concentration of those ligand molecules in your vesicle. These are all mechanisms of endocytosis, taking things in, but very similar processes can be used for exocytosis or pushing things back out. And that is an introduction to transport across the membrane.